Hello everyone, this is Dhanashri from the Dr. Omex Research Labs. So, in this session, we are going to discuss about the understanding of differentially expressed result. So, as of CovDiff is giving us this result, which is the gene expression.diff file, which is our main output file. So, now let's understand what information is present in this file. So, basically, CovDiff is giving us this many columns. But out of that, the columns which is required for us, we are going to only keep that column and rest of column we are going to delete. So let's discuss one by one column. Here, if you will see, this is what a test ID. These are nothing but some identifiers which is usually generated by the CovDiff tool. And if you are going to search this ID in any one of the database, for now you will not get any information about it. So better to delete this column. And same about the gene ID as well. So I'm going to delete this column as well. Now here we are getting the gene column which contains the gene name, gene symbol, or usually a gene ID. Uh, this is what the genes which got expressed in your samples. And how many genes are there? You can see total 1591 genes are there for now with this data. Then the next column, it will show you the locus. So this is a location where your gene is expressed. So basically it specifies the genomic location of the gene, which is giving in a format of BED, browser extensible data format, which is a very popular format in the UCSC database, which is nothing but the chromosome location, colon, start coordinate of the base and the end coordinate. So this is how it specifies the location. Then next column is a sample 1 and sample 2. So sample 1, actually it's the name of the first sample, or you can say the condition which is being compared. This is often the condition like a control, because in a hyphen L of CovDiff also, if you remember, we have given the first control and then treated, which means that our biological plan or our analysis plan is a treated versus control. And sample 2 is nothing but what? It's a name of the second sample or a condition. So here we are interested to compare the expression of treated with respect to control. Then next column, that is a status column which is indicating the test status. So this is telling you the status of your experiment. So if you are getting here the no test, it means like there was not enough alignment for the testing, or like the test was not performed, usually due to the low read count or other filtering criteria. Okay means what? The test is successful. And maybe if you are getting the fail, it means the test is failed due to the error or maybe some statistical calculations. Now, next column is a value 1 and value 2. So, value 1 and value 2 is nothing but what? It's your normalized expression values of your control and treated respectively. Then, our main column, which is nothing but the log2 fold change column. So, basically, this log2 fold change column, it will tell you the change of expression between the sample 2 and the sample 1, which is nothing but the treated versus control. Now, if I just want to show you how this CovDiff is calculated the expression value, so let's take this example. Now our plan we have decided to take is the treated versus control. So equals to log. Then here we are taking the value 2 divided by value 1 comma to the base 2 and enter. So you can see this is what the exactly same value we are getting, and this is how the CovDiff is calculated the log2 fold change value. Then next column we are having, that is a test statistics. So basically this test statistics value from the statistical test which is used to compare the expression level between the sample 1 and sample 2. So that is completely up to you that you want to keep this value or not, but I'm going to delete it. Next column, and again the next important column other than the fold change is a p-value. So this p-value, it's actually the statistical test which is indicating the probability of error value. So it is a probability that observed difference, or you can say a more extreme difference, would occur by the random chance if there were no actual difference. So basically this p-value, it will tell you about the significance, that whatever the gene which is expressed in your data, it is significantly expressed or not. So as it is a probability of error, error should be less. And what is the cutoff for the p-value then? We want p-value less than or equals to 0.05, which means we are allowing the 5% error over here. Then next is what Q value. So Q value, actually it's a adjusted p value. You can also say it's an adjusted p value after checking the false discovery rate, which accounts for the multiple testing. So basically this helps control the expected proportion of false positive among the set of significant results. So here also the cutoff will be same. And the last column, it will tell you about the significance, whether the change in expression is considered significant based on the Q value threshold. So if yes, it means it is a significant, and if it's no, it means it's not significant. But we can tell the significant based on the P value and the Q value, so no need to keep this column. So that's why I'm going to delete it. So this is what our final required columns. Now this result, I'm going to save it into the Excel workbook format into the RNA-seq folder. You can give the name as a D result or whatever name you want to give. You can just save in Excel workbook format. Now here, if you will see, whatever the genes which we are having over here, they are not significant. They all are not significant. So now the first step in the analysis, that we want to filter out the significant genes here. But before filtration of significant genes, if you will see that, like for this first column, we are having the multiple IDs at some rows. And these are nothing but the isoforms only. Isoforms are nothing but what? These are the multiple transcripts which are originated from the same locus. You can see the location is same, it's just the transcripts are different. So we can't keep a multiple IDs at a single row. Either we need to keep it below, otherwise we need to discard. But when to keep and when to discard, how can we decide that? So basically, if these IDs are coding for same gene, so at that time you can discard it. But if it is coding for different genes, at that time you can keep it below. Because if they are overlapping, obviously they will show you the same gene, right? So no need to keep that because the annotation will be same then. But how to check whether they are coding for same gene or for different gene? So as these IDs are from NM, like it is showing the prefix NM, which is nothing but the NCBI IDs. So you can check it into the NCBI database. But maybe if it is, sometimes if you are taking the reference genome and the GTF file from any another database, so according to that database, you can check. So I'm just going to copy this first ID here. And let's go with the NCBI gene database, and I'm just going to paste it. Then second ID also, I'm just going to paste it over here. And here I'm going to paste the third ID also, so that we can compare. Hmm. So if you will see the first, it is showing for some MPG gene. The second also it's for the same gene, and third it is also for the same gene. Which means that these IDs are having the same genes, so you can discard it. And one more thing, no need to check for all. If you just want, you can check it for one and two row, or like 2 to 3 rows, and if it is following the same pattern, you can directly discard it. So this for the discard I will show you, but just consider what if it is showing for like what if it is showing the different IDs. So at that time, how can you keep it? Then you just have to copy this complete row and you have to paste it below. In case if it is showing the different IDs, at that time I'm just showing you, and here I'm pasting it 2 times. Now from this, I'm just going to remove the first ID because we are already having the first ID above, and then third ID I'm going to remove from here. And from this, I'm just going to remove the first 2 IDs. And then from this row, I'm going to delete the last 2 IDs, so that first ID will be here and last 2 IDs will be there. So this is how you can do, but manually it is not possible. I can understand. So you can take the help of some Excel formulas or some programming language for it. But as of now, in our case, all the IDs are same. So we have to discard it. And how to discard it? You just have to copy the A column. Then you can add the new sheet, paste it over here. And as these IDs are separated by comma, so you can go with the data, text to column, then delimited it, separated based on comma, next and finish. Now I'm just going to copy this first column, go back to the previous sheet, and I'm just pasting it over here. So this is how we are having a one row in one, sorry, we are having the one ID in one row. Now once we are having this, we can filter out the significant genes. But again, before filtering the significant genes, I just want to apply the color filter here for the log to fold change column, just to see that how we are getting the upregulated genes, or based on the fold change gradient, we can differentiate the upregulated and downregulated genes. So for that, here we are going with the home, conditional formatting, new rule, and then here as we are having a 3 values, so we are selecting the 3 color scale. So we are getting minimum, midpoint, maximum. Minimum, that's for the negative value, which is a downregulated. Midpoint, that is a not expression value. And the maximum, these are the positive value, which means a upregulated. So I'm selecting here the number, and what is the cutoff for the downregulated? We are giving the -1. Then here for the not expression, 0, and for the upregulation we are giving the 1. For the downregulated we are giving the green color, for the not expression we are giving yellow, and for the upregulated we are giving the red color. This is a standard color. Instead of green you can give blue as well and say OK. So here you will see that based on the fold change gradient, we are getting the different shades of the color. So this is what a color filter we have applied for the fold change, and now we are going to filter out the significant genes. So for that we need to apply the filter on p-value column. So I'm selecting the p-value, go with the data, filter, then number filter, less than or equals to 0.05. Say OK. Now select the complete data and you can paste it here in the new sheet. You can rename this sheet as a significant genes, where p-value less than or equals to 0.05. Now you can go back to the previous sheet and remove the filters so that you can have your original data in the first sheet and your filtered data in the next sheet. So how many significant genes we are having over here right now? You can see 105 genes total. Now, as we got the significant genes, but still we are not getting any annotation because we are not having any gene symbol, or we don't know in which molecular function it is involved, what is the biological process, what are the cellular components, in which pathway it is involved. So for this, we need to retrieve the annotation. So that will we are going to discuss in our next part. Till then, if you have any question, please let us know. Bye. Have a great day.